All right. So do you guys want me to get started?
Yeah, let's go ahead and do it. So everybody, tonight we have Dr. Aspen Clements Miller as our speaker. She just finished her PhD in July of this year at the University of Virginia. And now she is teaching chemistry at Madison County High School in Virginia as well. So she's an alum of UNK from the chemistry department, graduated in 2015, so fairly recently. So her talk tonight is about the origins of life and perhaps where some of this came from. So Aspen, take it away.
Yeah, so when asked to give this talk, I thought, what would be the hardest topic for me to cover in the smallest amount of time? So I went with origins of life, which is great. And so just a little background about myself. So I got my PhD at UVA and I focused on theoretical astrochemistry. So really that's talking about the formation of molecules in different environments, let it be, in protoplanetary disks or molecular clouds, which I will get into what that all means in a little bit. So the grand old question of where did we come from? That's kind of where I wanted to start talking about. So talking about more about the ingredients instead of about how they could have formed, because that's an entirely other number of subjects, really, many PhDs. So when looking about this topic, I kept thinking, how do I go about talking about this? And I thought really the best place to start would be how we define life. So I found this on Wikipedia from John Casti, who was a mathematician, and it's pretty simple. It's an entity is considered to be alive if it has the capacity to carry out three basic functional activities: metabolism, self-repair, and replication. And so, if we're talking about the coronavirus, for example, we can see why it would not be considered alive, because it doesn't have the ability to replicate for itself. It has to use a host. So it's kind of helpful when we're trying to draw lines between what is alive and what is not. All right, so my field is astrochemistry, and that's looking at various objects and looking at the chemistry that occurs within them. And when talking about this, you really can't escape the astrophysical questions that a lot of people ask, like how solar systems are created. And so this is just the evolutionary stage of a giant molecular cloud, which is a bunch of gas and dust that's diffuse, and then it eventually can collapse upon itself, and it will form a disk and a protostar that then can evolve further and start to form little planetesimals, comets, and things of that nature. And then the dust and everything between starts to spread out, and you have your full-blown sun with all of the evolving planets. And so we have a pretty good idea of this is how the stages go. Of course, we have questions about what's happening in the accretion disks and things like that, and more of an astrophysical viewpoint. But as a chemist, we ask ourselves, so we have biological molecules or pre-biological molecules, and where are they coming from? Are they being formed on planets? Are prebiotics being delivered to early Earths and then something happens that causes biological molecules to form? Or did we inherit biological molecules from this disk, or maybe even the molecular cloud? So where does our chemical complexity really start? And so that's the question. Where did biological molecules form? Oh, I did mix that up. I said form and I said from for some reason. I messed up my typo. Anyway, so going back to our little view, there are many things that we've observed. So we've seen things like formaldehyde, which we don't really think of that as a prebiotic molecule, but it could be considered one. Glycoaldehyde, which a lot of people call a sugar, but it's actually technically not a sugar. It is basically the next simple molecule away from a sugar. And these have all been observed, and we can also make them in the lab and see how they are formed. But that doesn't really answer our question of what about biological molecules? So we have to keep expanding our scope. So talking a little bit about the prebiotic molecules. So this is a lot of research that I actually focused on, is taking your simplest components, like something like CO, which is a very abundant molecule, and hydrogen, which is the most abundant atom, and seeing what happens when you put them on a surface. So if you have CO and H, you can actually form things like formaldehyde, like we just talked about, and glycoaldehyde. But we can also form things like glycerol, and then potentially, I don't know if anyone's done this yet, because this gets to be quite a mess because you can get a lot of other products, but you could form something like ribose, which is really important for metabolism and for the structure of a lot of biological molecules. And just as an idea, so I talk about hydrogenation on surfaces. Let me go back. I wanted to mention this. That's not back, that's forward. There we go. So in the molecular cloud, there are things called grains, and on grains, it's basically acts like a catalyst. So you can have things accrete onto the surface and react. And so that actually kind of comes up again in the future when we start talking about biological molecules again. We have surfaces that can act like catalysts, which are really important when we're talking about forming complex molecules. All right, so there's something called the foremost reaction, which can take our simple molecules that we have seen, like formaldehyde and glycoaldehyde, and you can follow a couple chemical steps and form ribose. I don't think we really know exactly how this is formed, but there is cause to believe that something is happening to have it be made on comets, which I'll talk about in a second. So we still don't quite understand the mechanism to form ribose, but we have seen it, not in our molecular clouds, but actually in comets. All right, so getting into actually making biological molecules. So we've seen that we have some relatively complex species. And when I say that, I mean that from the astrochemical standpoint, because they're not biological, they're not complex, they're actually quite simple. But we want to get into how we can make our simplest components of our biological molecules. So this was actually a paper that was just released today in Nature Astronomy. And that's me. Sorry, this was very exciting for me. So we looked at a non-energetic mechanism to form glycine in ices. And so I was talking about in the interstellar medium, or in the molecular clouds, you have surfaces. And on that surface, you can actually have the formation of glycine. While it's not extremely abundant, we are still seeing glycine being formed in the earliest stages of solar systems, which is really exciting. We don't necessarily know how all of these things are inherited from one stage to the next, but it's kind of a way to look forward and see if maybe this is part of the reason we have glycine on early Earth and, finally on Earth itself. All right, so we've been able to make ribose doing irradiation of interstellar ices. So maybe this is a way that ribose is formed. It's not entirely clear, but we know that we have a lot of ultraviolet irradiation from stars. So this is a potential formation route. So then, okay, we have a couple formation routes. Well, what about in sources that we can actually, instead of using a telescope, we can actually get samples from? And what I mean by that is comets, because I think it was the year 2014 with the Rosetta mission was the first time that we ever landed actually on a comet, which was a really exciting and big deal. And we were able to find out a lot about comets from that. So we were able to find so this is a meteorite that actually landed on Earth, and we were able to detect nucleobases from it. And so nucleobases, hopefully you'll remember that this is part of what makes up RNA and DNA. So here's a little depiction, I think of this is uracil. And so this is part of the part that basically keeps our information. And if we remember going back to our definition of life, we needed to keep in mind how we store information and then how we also pass that information on. So we've seen that we can detect nucleobases. All right. We've also seen that we have amino acids and phosphorus. Phosphorus is another really important part of DNA and RNA. So far we were able to find all these ingredients that we for sure need. So this is the comet that we landed on in 2014. People always call it 67P CG just because it's an incredibly hard name to pronounce, at least for me. So this is exciting. We know that we have prebiotics on comets that have been potentially delivered to early Earth. We've also seen, and this was a new one for me, I actually hadn't known that we had ribose found on meteorites. So we have sugars as well. So we had glycoaldehyde in our molecular cloud, but then we've also seen it in things like meteorites, which is really exciting. And I'm not going to talk about this in this talk, but another really important thing to notice is when you look at comets, you'll notice that they have handedness or chirality. And so you'll see that they all have D configurations, which is interesting because that's what we see in biological molecules. And we don't really know where that comes from. We don't know why they specifically are D. So that's a whole, that was actually what my PhD was going to be on. And then I found out that was way too hard to try and do that theoretically because it's quite a complicated story. So we have all of our components in various sources. So how did they actually get delivered to Earth? That's the question. And so in your solar system, in your early solar system, you have a lot of chaos. Your planets are kind of moving around trying to find basically their gravitational equilibrium, you could say. And what can happen is you can have a lot of collisions between planets and things like little planetesimals, or even like what can become satellites and things like that. So this is, in my opinion, probably the most likely cause of how we got a lot of things like water on Earth and a lot of prebiotics, potentially actual biological molecules such as ribose. All right. So this is a depiction of early Earth. So in early Earth, there was pretty moderate temperatures, depending on what stage you're talking about. But there is, you can look in here, there's a lot of water. And within this water, we think that this was kind of where the biological spark happened, because you have a lot of material being able to kind of come in and come out, right? And so if you didn't have water as a medium, I don't think it would have been able to happen, which is why a lot of people focus on finding water in other bodies to see if we can find life. So within these bodies of water, you can often have things like, and there's a couple of different ways people think life could have formed. So it could have been a lot of heat from volcanoes. It could have been irradiation from the sun. There has to have been something that was energetic enough to kind of push life along. So we kind of get this whole idea of our primordial soup. So we have all of the materials, and we're basically just needing to start cooking it. So that's kind of the theme of this talk was, so we have our ingredients, how do we make our soup, which then potentially will lead to life. So this is a very exciting experiment that actually got me into astrochemistry to begin with. I learned about this in biochemistry. So this is the Urey-Miller experiment, and it happened in 1952. Essentially, Urey and Miller took a lot of basic compounds, so methane, water, ammonia, H2, and then shot a shock through it and then just condensed it and collected the sample. And what they found was a number of amino acids. And this experiment has been done time and time again with various tweaks, like you might want to add sulfur, for example, or phosphorus, just because those can be important for biological molecules. And as time has progressed, our detection methods have progressed, and we've been able to see that most of the amino acids that are important to us actually form in this experiment. So another thing, you can also make sugars. All right. So the reason I mention this is there's kind of a couple different opinions. So some people believe that life, not life itself, but biological molecules formed on Earth. Some people believe that it was delivered to early Earth. Probably it's going to be a combination of things, just like everything. It's a little more complicated than one over the other. But it's just interesting to see that our most simple components can be turned into what we consider the most complicated structures, like what we are made of. So this is a bit of an advanced Urey-Miller experiment. So this was done from NASA Ames, and they took Lyman alpha, which is this pink color. It's a hydrogen line, and they irradiated pyrimidine in water, and they were actually able to get uracil, cytosine, and thymine. So these are all three components of RNA and DNA, which is important, again, because we're talking about life and how we're trying to maintain this code to progress life and potentially mutate it and evolve. And then here's just a little picture. So in this little vacuum chamber, you would have an ice, and then you would have your water molecules. And this is not to scale. This was just a little doodle on from me just to clarify what was going on. And something to note, because I was thinking about this. So we have pyrimidine, which is a ring structure. And I was thinking, okay, have we seen ring structures actually? Have we observed them in interstellar medium or in molecular clouds? And there was actually a recent paper published from a group at UVA who saw the first ring structure, and they found benzonitrile. And the thing about benzene in particular, so if you basically just remove this CN group and add a hydrogen, the problem with it is it's incredibly hard to detect because it is symmetric. So you have to have some kind of pole in a specific direction to easily detect it. And they're doing this in radio wavelengths, if anyone in particular is curious. So the issue with thinking about how we find ring structures and things is that we have to deal with the fact that we are looking, one, at complicated molecules, which makes it harder to detect, but also we have to make sure that we have dipole moments. So in case you were curious about if we had seen something like pyrimidine, we've started to kind of open our horizon into being able to find benzene rings and potentially things like even buckyballs, which is another topic. But it's just complicated because you have to worry about all of the different energetic modes and how you'll start to get overlap with other molecules. So it's a kind of a challenge in itself. So the real question is, so we have all of these ingredients. All right, that's cool. So how do we actually transition from biological molecules to biology? And this is the part where I'm no longer as good at this, just because I'm trained more in chemistry, but I think it's really important and it's also very interesting. So there's a couple different theories actually as to how how life itself, or the first cell could have been formed. So I'm going to only mention a couple of them because I think there are plenty out there to read about. But so there's some theories as to thinking that metabolism was actually the first growth. So metabolism led to macromolecules. And so I read a paper about this. They were talking about the Archean Ocean. So there's no oxygen. It's a pretty warm climate. And what they found was there was actually metabolic like reactions occurring. And so the people who did this paper, I think I put it in the, nope, I did not. I'm sorry. I hope I had the title, but I did not have it, but I have it up in my, my internet browser. But so what they did was they set up early Earth-like conditions. That's up to speculation, right? We don't necessarily know exactly what it was like, but let's assume that they did a good job. All right. They were able to actually form this structure. And this structure, I'll go to the next slide, is extremely important because it leads to RNA and DNA. So it's the phosphate and the deoxyribose sugar. So from these early Earth conditions, which were just leading to metabolic-like reactions, we were able to form the backbone, essentially, of RNA and DNA. And this is actually, I was reading quite a complicated process. So RNA is kind of tricky. It's easy enough to form, the nucleobase and the sugar, but then trying to get everything to piece together is quite a tricky business. So if we ignore that part and you go into the actual structure, RNA is quite interesting in that it can actually form as a catalyst for itself. It can start to form, it can start to add other nucleobases and do self-replication, which is really interesting, which is why people also thought maybe it was more of an RNA world hypothesis. So instead of metabolism coming first, maybe it was RNA, or maybe they're kind of mixed together. So in this hypothesis, RNA is the first thing that came to be before, because we don't really know if DNA came first or RNA came first. But it would kind of make sense that RNA came first because it codes for proteins and DNA, and also it is able to self-replicate. The only issue, again, though, that I was talking about was that trying to get it onto its backbone. If we can get it onto its backbone, it seems like RNA is quite the candidate for this process, but we don't know that step yet. So, all of this to say, it appears that we have the ingredients, but what is the recipe? So we found that we have nucleobases in comets, we have ribose in comets, and we also have amino acids in comets. But once it gets to Earth, how did we form it? And there's a number of questions that still need to be answered, like how do membranes form? Because once you do have something like a cell, how do you close it off so you're not opening it to contamination or immediately destroying it? So this is a huge study or field of study. I know that I couldn't go into great detail about a lot of stuff, but I was hoping just to talk briefly about a wide range of things. And I was really hoping we could have a good conversation about all of this. Thank you.
Alan, I'm seeing that you have a question here. You can go ahead and unmute yourself. Alan, are you there? Sorry, he doesn't have a microphone. So who has questions here? Feel free to unmute yourself, ask away.
Can you hear me?
Yeah, go for it, Brad.
Okay. Have you read any of Robert Hazen? Or are you familiar with Robert Hazen?
I'm not.
No. He's done a lot of work along these lines. He's shown a number of ways in which you can derive these molecules by various processes, whether it's UV irradiation or high impact, or a number of routes. So it's always been amazing to me as a molecular biologist just how easy it is to make these molecules, and not just by one route. I noticed, however, that you just have pyrimidines, no purines. So would you care to comment on production of purines?
So interestingly, I kept seeing pyrimidines over and over again, and I didn't actually see much about purines, and I didn't even like really go into it that much.
Okay, well, I think if I'm remembering his discussions properly, that cyano compounds under the right conditions will at least form adenine. Guanine seems to be a little trickier to make, but you can at least get adenine via relatively simple processes. And then your comment concerning lipids. What's the name of that asteroid they found in Australia? Very famous, actually. I can't believe I can't think of it right now. But anyway, when they took samples of that and ground it up and did an extraction for lipids, voila, those lipids that they extracted are able to form vesicles. So the molecules necessary for membranes are also out in outer space.
Yeah, I didn't go too much into membranes because I already felt like I was pushing my limits with what I could talk about comfortably in 30 minutes. So, but yeah, no, it is really fascinating, and I did a little bit of research in like surfactants, and it was quite fascinating that that's just such a. I mean, it makes sense, right? Everything in biology probably mimics something that's going to be physically reliable. So it makes sense. But yeah, it's fascinating stuff that it's kind of everywhere too. And of course, it's carbon and nitrogen and oxygen that we're made of, because that's what we have abundance of.
I was kind of curious about, you talked about the heavy bombardment phase of like an early solar system development. And I'm kind of curious, how well do these molecules hold up to that kind of stress? I mean, are they, so they're being, so hypothetically they're deposited from, say, a comet onto a forming planet. When that impact happens, is that going to destroy any of this stuff?
Yeah, so when you have a comet coming in, if you have any kind of atmosphere, it's going to tear up a lot of the ice and vaporize it before it even impacts. And then, I mean, I'm not sure quite about the physical processing of it, but I'm sure you lose a ton of material as it's approaching the object, especially if it's has a significant atmosphere. Yeah, that's an interesting question. I don't know about breaking the molecules themselves apart. There might be some kind of, as they're heated, because a lot of these things are in ices. So if you're heating it, there's also some things that could be happening to the ices themselves. So I think there's some concern about, my advisor is actually doing a model on comets and looking at, you can have a comet and this side won't be facing the sun and this side will. So what kind of gradient are you going to have based on that? So there's a lot of science that's still being looked into, because we don't really know that much about comet processing, because we're talking really thick ices. How much is being processed at the very surface versus the very deep layers? So that itself could complicate that question significantly.
Yeah, I guess I was just thinking, because you were talking about things forming on surfaces. And so I was like, well, would they be inside of it? Or, I don't know how that works.
Inside of?
Like inside of your comet, right? Like would any of these molecules be found within it or just on the outer surface?
Yeah, so it's kind of interesting because it's comets, as I understand it, they're based of a bunch of like little rocky pebbles, and then they're kind of iced together as well. And then there can be an accumulation of rocks kind of in the center as well. And this is more into the formation of like planetesimals, so little tiny planets. So I know that for them to form, you basically need that icy stuff to kind of get it to stick. So there will be material kind of throughout the comet itself. It's not just like a rock in the center and then ice. It's usually kind of a mix all around. So that's why I think chunks of it would be able to break off as it's coming through the atmosphere.
Hey, Evan, do you know if Ken Trantham's with us today?
It does not look like Ken is with us here, unfortunately.
Well, the reason I bring it up is some of his work, and because this is like third hand or at best, he was doing some stuff with spin polarized electrons, and he said in the universe there is an abundance of one type of polarization, and what he was looking at was preferential destruction of one-handedness, and that be a potential cause for the reason what we would see mostly the D.
Yeah, there's definitely, so I think that's the main theory at this point, is that it's polarized light that has this effect. But I don't really know much about it. I know what
he was doing was actually spin polarized electrons, and so you've got all the beta particles flying around, and certainly there'd be plenty of those as well as the light. So one more mechanism perhaps.
Yeah, well, because I was gonna try and do a program with this, but then it was gonna basically be not a rate equations model, but something similar to that. And you're gonna have to take into account each handedness of each molecule. So it becomes a pretty nightmarish scenario for a modeler pretty fast. But yeah, that's very interesting. I'm curious to talk to him about it sometime.
Can you hear me?
Oh, yep.
Oh, sorry. I really appreciated that you reviewed the more in depth the RNA world model, because like anything I've read is basically implicated that however life got here, it involved RNA at some key point, like that was one of the key steps. And so you talked about how like that particular part of getting the sugar on the phosphate backbone was sort of like the stuck point in the research right now. So I guess like my question was, do you know like the people who are pursuing that, who are trying to resolve that, like what are some of the techniques that they might use, or what are some of the ways that they might try to help us figure this out?
Yeah, so there's a couple different theories actually, because of course within one theory there's like 5 more theories. And I was reading that instead of having a phosphate backbone, it was thought that it could have been some kind of inorganic catalyst, essentially. So something like zinc or iron, kind of like maybe a clay material, which I found quite fascinating because of given kind of the nature of what I was doing, which was using silicates like sandy material as like a grain to allow things to accumulate. So from what I honestly, this is very much not my expertise, but what I was reading was that there were theories that there was some kind of inorganic catalyst that might have been the process, and then it was slowly moved out for the phosphate group. But I don't really know what research goes on and how they go about doing that.
That is more than I knew before, though. So thank you.
Yeah, it's quite fascinating stuff. But like I said, it's just such a broad topic. There was a researcher who came in and started talking about the formation of membranes, and basically just putting stuff. I mean, I'm sure there was a lot more to it, but putting stuff into a vat and just to see like the structures that they form, and being able to see like kind of what we were talking about before, were like vesicle formation, and like surfactants, how they can form like a little membrane of sorts. I think it's a lot of, you put something in and you observe what happens. You just have to try and get the right ingredients inside of it.
The name of that meteorite, by the way, was the Murchison meteorite. So
This guy. I wondered if that was the one you were talking about.
Right. Any other final questions here? Going once, going twice. I'm going to guess that's about it. Aspen, thank you so much for giving this talk today. It's a fascinating topic, and there's, like you pointed out, there's so much out there that has to go exactly right, in order for all of this to happen, and we certainly don't know. But really enjoyable. Thanks so much for taking the time this evening.
Thanks for having me. It was really fun.
You bet. Always great to have a UNK alum on here. Hopefully we can get you to come back. Well, I don't know, maybe you've already done 3 for us, so we'll give you a couple of years off.
Well, I do think I'm going to go back for the research symposium sometime in person, which will be great. So maybe then.
Awesome. Yeah, I think we're all really looking forward to getting back to doing things in person again as well. So let's keep our fingers crossed and hope that happens soon.
I'm glad to see UNK's Sigma Xi group is going strong.
We're doing all right. Yeah, we're holding it together. This term was a little bit of a challenge, right? But I think we've got a good base. I think we're going to figure it out and at least be able to keep doing what we're doing right now in the spring, and maybe that vaccine will come along and we'll be back in person next fall.
Well, I kind of love how not to keep dragging this on, but I love that it allows people to give talks who potentially wouldn't have been able to give talks. You can really spread your net in a way.
Absolutely. Well, when we first met as an executive committee, Dr. Carlson asked Fauci if he would be a speaker for us, and turns out he was busy.
Yeah, probably will. But you got to try.
Yeah, exactly. So, yeah, it is nice to be able to have people, from Virginia, for example, come in and give a talk as if they were here. That is very, very convenient.
Yeah, it is. Brad, thanks I hope that it keeps going. Did you see Moderna, the vaccine yielding 94.5% efficacy? That's
better than a number of current vaccines, by the way.
How far has this one gone through? Where is it in the stages right now?
This is a report from phase 3 clinical. So they're now going to be requesting emergency release within the next few months.
Wow.
I think they said they only need like 35 more cases of somebody catching COVID in the group to have their statistics to get the emergency authorization. So
Yeah,
since
that's right.
it's actually interesting. They said that because we're having such a bad spike right now, it's helping the research go faster.
Yeah.
Yeah, it's I'm sure it is. I actually know a couple people that are in that study. So,
Silver lining, I suppose.
Well, good luck to them in the study. Good luck to everyone.
Yeah. Let's see here. All right. All right, everyone. Well, again, thank you all for coming. Aspen, thank you very much for this talk. It was excellent. And we'll catch up with everyone in the spring. This is our last one for this term. I think we'll have a couple more than we did this term in the spring. So Aspen, we'll get you info on those. You're free to attend.
All right. Bye, guys. Thank you.
thank you.